Toxoplasma Gondii by Virginia Knight. Toxoplasma Gondii is a protozoan parasite that causes the infection known as toxoplasmosis in warm-blooded animals. The typical host for T. gondii is domestic cats and other members of the Felidae family and is typically spread through cat feces. Other intermediate hosts for this parasite are birds and rodents. Animals typically pick up the parasite from consumption of oocyst-contaminated soil, water, or plant material. Oocysts are shed for approximately 1 to 3 weeks and take 1 to 5 days to sporulate, become infective, and transform into tachyzoites shortly after ingestion. The tachyzoites then implant in neural and muscle tissue and form parasiforous vacuoles. Once implanted, the tachyzoites then become bradyzoids and form tissue cysts. During this time in the host cell, the parasites steal nutrients to ensure proliferation and persistence. This picture from the CDC shows the infection cycle of T. gondii. Cats ingest any of the 3 infectious stages of the parasite and parasitic shedding begins. The shedding period and frequency vary among the 3 infectious stages. After ingestion, the bradyzoite cyst wall is dissolved by proteolytic enzymes in the stomach and small intestine. Bradyzoites then penetrate the small intestine epithelial cells and initiate development of T. gondii progeny through asexual reproduction and development. After asexual development, the sexual cycle is started by fertilization, which then forms an oocyst wall around the parasite. Infected epithelial cells rupture and release the oocysts into the intestinal lumen, which is then excreted in the feces. Intermediate hosts pick up the parasite after ingesting contaminated soil, water, or plant material. Oocysts transform into tachyzoites after ingestion and localize in muscle and neural tissue. A tissue cyst called bradyzoite then forms. Humans become infected in 4 ways: consumption of undercooked meat containing bradyzoites, consumption of water contaminated with infected cat feces or environmental samples, blood transfusions or organ transplantation, or transplant centellate from mother to the fetus. In humans, T. gondii infection can be found in skeletal muscle, myocardium, neural, and ocular tissues. Cysts can remain in these cells for the rest of the human's life. Toxoplasma Gondii infection can be diagnosed through highly specific and sensitive molecular testing methods. These methods include microscopic diagnosis, bioassay, serological assays that include dye testing, modified agglutination testing, latex agglutination testing, indirect hemagglutination testing, indirect fluorescent antibody testing, enzyme-linked immunosorbent assay, immunosorbent agglutination testing, immunochromographic testing, piezoelectric immunoagglutination assay, Western blot, and avidity tests. Imaging techniques include computed chromatography, magnetic resonance imaging, and ultrasonography. Molecular methods based on detection of Toxoplasma Gondii nucleic acids include PCR, real-time PCR, and loop-mediated isothermal amplification. Genotyping methods based on molecular technologies include microsatellite analysis, multilocus sequence typing, PCR RFLP, random amplification polymorphic DNA PCR, and high-resolution melting. The picture on the right is an MRI of someone's brain who has been infected with Toxoplasma Gondii. The arrows pointing to the white dots are plaque formations that result from Toxoplasma Gondii infection. When the parasite first forms the parasiforous vacuole or tissue cyst, it uses stolen lipids from the host cell to form a permeable layer that can transport molecules freely. The vacuole is then modified by T. gondii proteins to interfere with protein export and importation of lipids and polar metabolites. The parasite is able to transport vitamins, sugars, amino acids, nucleobases, nucleosides, and nucleotides into the vacuole. However, the required nutrients T. gondii needs from the host cell depends solely on the availability of other nutrients and environmental conditions. T. gondii uses glucose and glutamine as its major carbon sources and uses acetate obtained from outside the cell to elongate fatty acids. This parasite requires tryptophan from outside the cell and also requires arginine and tyrosine. Alanine, valine, leucine, isoleucine, proline, glycine, serine, threonine, methionine, tyrosine, and phenylalanine are all important to the parasiforous vacuole for the parasite to use. The parasite uses purines obtained from nucleic acid and imports vitamins that are used in enzymatic reactions. While the pyrophorus vesicle is advantageous by protecting T. gondii from acidification, essential nutrients can't get inside the vesicle. To combat this issue, there are small pores in the PVM that allow small molecules to pass through. This passageway is independent of temperature and energy, which affords the parasite an efficient bidirectional molecular exchange mechanism within the host cell. T. gondii also utilizes active transport through its plasma membrane and the PVM to obtain metabolites attached to large proteins that are unable to enter the cell through the diffusion. Proteins acquired through the development of the PVM are most likely acquired from the release of the bulbous part of rhoptries during the parasite's invasion into the cell. The PVM proteins are involved in nutrient acquisition, but is also used to recruit host mitochondria and endoplasmic reticulum that are used to transfer nutrients to the parasite. Infected patients are often prescribed a drug cocktail of pyrimethamine and sulfadiazine with folinic acid. Pyrimethamine is a diaminopyrimidine that is effective against the erythrocytic stage of T. gondii that inhibits plasmodial dihydrofolate reductase. Sulfadiazine is a derivative of sulfanilamide, which interferes with the folic acid synthesis by inhibiting dihydroputyroate synthase. This enzyme is involved in folic acid synthesis that precedes the step backed by pyrimethamine. Folinic acid is essential because it decreases the incidence of bone marrow suppression that is caused by pyrimethamine. Thank you for watching my video. I hope you learned some interesting things about Toxoplasma gondii.